Gene therapy. Gene therapy is a medical field which focuses on the genetic modification of cells to produce a therapeutic effect or the treatment of disease by repairing or reconstructing defective genetic material. The first attempt at modifying human DNA was performed in 1980 by Martin Cline, but the first successful nuclear gene transfer in humans approved by the National Institutes of Health was performed in May 1989. The first therapeutic use of gene transfer, as well as the first direct insertion of human DNA into the nuclear genome, was performed by French Anderson in a trial starting in September 1990. It is thought to be able to cure many genetic disorders or treat them over time. Between 1989 and December 2018, over 2,900 clinical trials were conducted, with more than half of them in phase I. As of 2017, Spark Therapeutics Luxturna RPS65 mutation-induced blindness and Novartis Kymriah Chimeric antigen receptor T-cell therapy are the FDA first approved gene therapies. Since that time, drugs such as Novartis Zolgensma and Alnylam's Patisiran have also received FDA approval, in addition to other companies' gene therapy drugs. Most of these approaches utilize adeno-associated viruses AAV and lentiviruses for performing gene insertions, in vivo and ex vivo, respectively. AAV are characterized by stabilizing the viral capsid, lower immunogenicity, ability to transduce both dividing and non-dividing cells, the potential to integrate site-specifically, and to achieve long-term expression in the in vivo treatment. Gorell et al. 2014. ASO/cRNA approaches such as those conducted by Alnylam and Ionis Pharmaceuticals require non-viral delivery systems and utilize alternative mechanisms for trafficking to liver cells by way of GalNAc transporters. The concept of gene therapy is to fix a genetic problem at its source. If, for instance, a mutation in a certain gene causes the production of a dysfunctional protein resulting usually recessively in an inherited disease, gene therapy could be used to deliver a copy of this gene that does not contain the deleterious mutation and thereby produces a functional protein. This strategy is referred to as gene replacement therapy and is employed to treat inherited retinal diseases. While the concept of gene replacement therapy is mostly suitable for recessive diseases, novel strategies have been suggested that are capable of also treating conditions with a dominant pattern of inheritance. The introduction of CRISPR gene editing has opened new doors for its application and utilization in gene therapy, as instead of pure replacement of a gene, it enables correction of the particular genetic defect. Solutions to medical hurdles, such as the eradication of latent human immunodeficiency virus HIV reservoirs and correction of the mutation that causes sickle cell disease, may be available as a therapeutic option in the future. Prosthetic gene therapy aims to enable cells of the body to take over functions they physiologically do not carry out. One example is the so-called vision restoration gene therapy that aims to restore vision in patients suffering from end-stage retinal diseases. In end-stage retinal diseases, the photoreceptors as the primary light-sensitive cells of the retina are irreversibly lost. By the means of prosthetic gene therapy, light-sensitive proteins are delivered into the remaining cells of the retina to render them light-sensitive and thereby enable them to signal visual information towards the brain. Clinical trials are ongoing. In CT 2,556,736, in CT 3,326,336 at clinical trials. Not all medical procedures that introduce alterations, bone marrow transplantation, and organ transplants in general have been found to introduce foreign DNA into patients. Background: gene therapy was conceptualized in 1972 by authors who urged caution before commencing human gene therapy studies. The first attempt, an unsuccessful one, at gene therapy, as well as the first case of medical transfer of foreign genes into humans, not counting organ transplantation, was performed by Martin Cline on 10 July 1980. Cline claimed that one of the genes in his patients was active 6 months later, though he never published this data or had it verified, and even if he is correct, it's unlikely it produced any significant beneficial effects treating beta thalassemia. After extensive research on animals throughout the 1980s and a 1989 bacterial gene tagging trial on humans, the first gene therapy widely accepted as a success was demonstrated in a trial that started on 14 September 1990, when Ashanthi de Silva was treated for AAVs, the first somatic treatment. The goal was to cure malignant brain tumors by using recombinant DNA to transfer a gene making the tumor cells sensitive to a drug that, in turn, would cause the tumor cells to die. The polymers are either translated into proteins, interfere with target gene expression, or possibly correct genetic mutations. The most common form uses DNA that encodes a functional, therapeutic gene to replace a mutated gene. The polymer molecule is packaged within a vector, which carries the molecule inside cells. Early clinical failures led to dismissals of gene therapy. Clinical successes since 2006 regained researchers' attention, although as of 2014, it was still largely an experimental technique. These include treatment of retinal diseases, Leber's congenital amaurosis, and choroideremia, X-linked SID, ADIS-SID, adrenoleukodystrophy. Between 2013 and April 2014, U.S. companies invested over $600 million in the field. The first commercial gene therapy, Gendicine, was approved in China in 2003 for the treatment of certain cancers. In 2011, Neovasculgen was registered in Russia as the first-in-class gene therapy drug for treatment of peripheral artery disease, including critical limb ischemia. In 2012, Glybra, a treatment for a rare inherited disorder, lipoprotein lipase deficiency, became the first treatment to be approved for clinical use in either Europe or the United States after its endorsement by the European Commission. Following early advances in genetic engineering of bacteria, cells, and small animals, scientists started considering how to apply it to medicine. Two main approaches were considered: replacing or disrupting defective genes. Scientists focused on diseases caused by single gene defects, such as cystic fibrosis, hemophilia, muscular dystrophy, thalassemia, and sickle cell anemia. Glybra treats one such disease, caused by a defect in lipoprotein lipase. DNA must be administered, reach the damaged cells, enter the cell, and either express or disrupt a protein. Multiple delivery techniques have been explored. The initial approach incorporated DNA into an engineered virus to deliver the DNA into a chromosome. Naked DNA approaches have also been explored, especially in the context of vaccine development. Generally, efforts focused on administering a gene that causes a needed protein to be expressed. More recently, increased understanding of nuclease function has led to more direct DNA editing, using techniques such as zinc finger nucleases and CRISPR. The vector incorporates genes into chromosomes. The expressed nucleases then knock out and replace genes in the chromosome. As of 2014, these approaches involve removing cells from patients, editing a chromosome, and returning the transformed cells to patients. Gene editing is a potential approach to alter the human genome to treat genetic diseases, viral diseases, and cancer. As of 2020, these approaches are being studied in clinical trials. Cell types. Gene therapy may be classified into two types. Somatic. In somatic cell gene therapy, SCGT, the therapeutic genes are transferred into any cell other than a gamete, germ cell, gametocyte, or undifferentiated stem cell. Any such modifications affect the individual patient only and are not inherited by offspring. Somatic gene therapy represents mainstream basic and clinical research in which therapeutic DNA, either integrated in the genome or as an external episome or plasmid, is used to treat disease. Over 600 clinical trials utilizing SCGT are underway in the U.S. Most focus on severe genetic disorders, including immunodeficiencies, hemophilia, thalassemia, and cystic fibrosis. Such single-gene disorders are good candidates for somatic cell therapy. The complete correction of a genetic disorder or the replacement of multiple genes is not yet possible. Only a few of the trials are in the advanced stages. Germline. In germline gene therapy (GGT), germ cells, sperm or egg cells, are modified by the introduction of functional genes into their genomes. Modifying a germ cell causes all the organism's cells to contain the modified gene. The change is therefore heritable and passed on to later generations. Australia, Canada, Germany, Israel, Switzerland, and the Netherlands prohibit GGT for application in human beings for technical and ethical reasons, including insufficient knowledge about possible risks to future generations and higher risks versus a CGT. The U.S. has no federal controls specifically addressing human genetic modification beyond FDA regulations for therapies in general. Vectors. The delivery of DNA into cells can be accomplished by multiple methods. The two major classes are recombinant viruses, sometimes called biological nanoparticles or viral vectors, and naked DNA or DNA complexes, non-viral methods. Viruses. In order to replicate, viruses introduce their genetic material into the host cell, tricking the host's cellular machinery into using it as blueprints for viral proteins. Retroviruses go a stage further by having their genetic material copied into the genome of the host cell. Scientists exploit this by substituting a virus's genetic material with therapeutic DNA. The term DNA may be an oversimplification, as some viruses contain RNA, and gene therapy could take this form as well. A number of viruses have been used for human gene therapy, including retroviruses, adenoviruses, herpes simplex vaccinia, and adeno-associated virus. Like the genetic material DNA or RNA in viruses, therapeutic DNA can be designed to simply serve as a temporary blueprint that is degraded naturally or at least theoretically to enter the host's genome, becoming a permanent part of the host's DNA in infected cells. Non-viral. Non-viral vectors for gene therapy present certain advantages over viral methods, such as large-scale production and low host immunogenicity. However, non-viral methods initially produced lower levels of transfection and gene expression, and thus lower therapeutic efficacy. Newer technologies offer promise of solving these problems with the advent of increased cell-specific targeting and subcellular trafficking control. Methods for non-viral gene therapy include the injection of naked DNA, electroporation, the gene gun, sonoporation, magnetofusion, the use of oligonucleotides, lipoplexes, dendrimers, and inorganic nanoparticles. More recent approaches, such as those performed by companies such as Ligandel, offer the possibility of creating cell-specific targeting technologies for a variety of gene therapy modalities, including RNA, DNA, and gene editing tools such as CRISPR. Other companies, such as Arbutus Biopharma and Arcturus Therapeutics, offer non-viral, non-cell targeted approaches that mainly exhibit liver tropism. In more recent years, startups such as Sixfold Bio, GenEdit, and Spotlight Therapeutics have begun to solve the non-viral gene delivery problem. Non-viral techniques offer the possibility of repeat dosing and greater tailorability of genetic payloads, which in the future will be more likely. Companies such as Editas Medicine, Intellia Therapeutics, CRISPR Therapeutics, KCBia Selecties, Precision Biosciences, Bluebird Bio, and Sangamo have developed non-viral gene editing techniques, however frequently still use viruses for delivering gene insertion material following genomic cleavage by guided nucleases. These companies focus on gene editing and still face major delivery hurdles. BioNTech, Moderna Therapeutics, and CureVac focus on delivery of mRNA payloads, which are necessarily non-viral delivery problems. Alnylam, Ionis Pharmaceuticals, and Ionis Pharmaceuticals focus on delivery of cRNA antisense oligonucleotides for gene suppression, which also necessitate non-viral delivery systems. In academic contexts, a number of laboratories are working on delivery of pegylated particles, which form serum protein coronas and chiefly exhibit LDL receptor-mediated uptake in cells in vivo. In vivo versus ex vivo therapies. Gene doping. Athletes may adopt gene therapy technologies to improve their performance. Gene doping is not known to occur, but multiple gene therapies may have such effects. Kaiser et al. Argue that gene doping could level the playing field if all athletes receive equal access. Critics claim that any therapeutic intervention for non-therapeutic/enhancement purposes compromises the ethical foundations of medicine and sports. Human genetic engineering. Genetic engineering could be used to cure diseases, but also to change physical appearance, metabolism, and even improve physical capabilities and mental faculties, such as memory and intelligence. Ethical claims about germline engineering include beliefs that every fetus has a right to remain genetically unmodified, that parents hold the right to genetically modify their offspring, and that every child has the right to be born free of preventable diseases. For parents, genetic engineering could be seen as another child enhancement technique to add to diet, exercise, education, training, cosmetics, and plastic surgery. Another theorist claims that moral concerns limit, but do not prohibit germline engineering. A recent issue of the journal Bioethics was devoted to moral issues surrounding germline genetic engineering in people. Possible regulatory schemes include a complete ban, provision to everyone, or professional self-regulation. The American Medical Association's Council on Ethical and Judicial Affairs stated that genetic interventions to enhance traits should be considered permissible only in severely restricted situations. 1. Clear and meaningful benefits to the fetus or child. 2. No trade-off with other characteristics or traits. And 3. Equal access to the genetic technology. With the advent of new techniques like CRISPR, in March 2015 a group of scientists urged a worldwide moratorium on clinical use of gene editing technologies to edit the human genome in a way that can be inherited. In April 2015, researchers sparked controversy when they reported results of basic research to edit the DNA of non-viable human embryos using CRISPR. A committee of the American National Academy of Sciences and National Academy of Medicine gave qualified support to human genome editing in 2017 once answers have been found to safety and efficiency problems but own. Treatment of genetic diseases. Gene therapy approaches to replace a faulty gene with a healthy gene have been proposed and are being studied for treating some genetic diseases. Diseases such as sickle cell disease that are caused by autosomal recessive disorders for which a person's normal phenotype or cell function may be restored in cells that have the disease by a normal copy of the gene. The risks and benefits related to gene therapy for sickle cell disease are not known. List of gene therapies for treatment of disease. Some genetic therapies have been approved by the U.S. Food and Drug Administration (FDA), the European Medicines Agency (EMA), and for use in Russia and China. Adverse effects, contraindications, and hurdles for use. Some of the unsolved problems include short-lived nature before gene therapy can become a permanent cure for a condition. The therapeutic DNA introduced into target cells must remain functional and the cells containing the therapeutic DNA must be stable. Problems with integrating therapeutic DNA into the genome and the rapidly dividing nature of many cells prevent it from achieving long-term benefits. Patients require multiple treatments. Immune response. Anytime a foreign object is introduced into human tissues, the immune system is stimulated to attack the invader. Stimulating the immune system in a way that reduces gene therapy effectiveness is possible. The immune system's enhanced response to viruses that it has seen before reduces the effectiveness to repeated treatments. Problems with viral vectors. Viral vectors carry the risks of toxicity, inflammatory responses, and gene control and targeting issues. Multigene disorders, some commonly occurring disorders, such as heart disease, high blood pressure, Alzheimer's disease, arthritis, and diabetes, are affected by variations in multiple genes, which complicate gene therapy. Some therapies may breach the Weismann barrier between soma and germline, protecting the testes, potentially modifying the germline, falling afoul of regulations in countries that prohibit the latter practice. Insertional mutagenesis, if the DNA is integrated in a sensitive spot in the genome, for example in a tumor suppressor gene, the therapy could induce a tumor. This has occurred in clinical trials for X-linked severe combined immunodeficiency (XSID) patients, in which hematopoietic stem cells were transduced with a corrective transgene using a retrovirus, and this led to the development of T-cell leukemia in 3 of 20 patients. One possible solution is to add a functional tumor suppressor gene to the DNA to be integrated. This may be problematic since the longer the DNA is, the harder it is to integrate into cell genomes. CRISPR technology allows researchers to make much more precise genome changes at exact locations. Cost allopogene to parvovic or glibra, for example, at a cost of $1.06 million per patient, was reported in 2013 to be the world's most expensive drug. Deaths. 3 patients' deaths have been reported in gene therapy trials, putting the field under close scrutiny. The first was that of Jess Gelsinger, who died in 1999 because of immune rejection response. One ex-SID patient died of leukemia in 2003. In 2007, a rheumatoid arthritis patient died from an infection. The subsequent investigation concluded that the death was not related to gene therapy. Regulations covering genetic modification are part of general guidelines about human-involved biomedical research. There are no international treaties which are legally binding in this area, but there are recommendations for national laws from various bodies. The Helsinki Declaration, Ethical Principles for Medical Research Involving Human Subjects, was amended by the World Medical Association's General Assembly in 2008. This document provides principles physicians and researchers must consider when involving humans as research subjects. The Statement on Gene Therapy Research, initiated by the Human Genome Organization (HUGO) in 2001, provides a legal baseline for all countries. HUGO's document emphasizes human freedom and adherence to human rights and offers recommendations for somatic gene therapy, including the importance of recognizing public concerns about such research. United States. No federal legislation lays out protocols or restrictions about human genetic engineering. This subject is governed by overlapping regulations from local and federal agencies, including the Department of Health and Human Services, the FDA, and NIH Recombinant DNA Advisory Committee. Researchers seeking federal funds for an investigational new drug application, commonly the case for somatic human genetic engineering, must obey international and federal guidelines for the protection of human subjects. NIH serves as the main gene therapy regulator for federally funded research. Privately funded research is advised to follow these regulations. NIH provides funding for research that develops or enhances genetic engineering techniques and to evaluate the ethics and quality in current research. The NIH maintains a mandatory registry of human genetic engineering research protocols that includes all federally funded projects. An NIH advisory committee published a set of guidelines on gene manipulation. The guidelines discuss lab safety, as well as human test subjects and various experimental types that involve genetic changes. Several sections specifically pertain to human genetic engineering, including Section Roman 3C-1. This section describes required review processes and other aspects when seeking approval to begin clinical research involving genetic transfer into a human patient. The protocol for a gene therapy clinical trial must be approved by the NIH Recombinant DNA Advisory Committee prior to any clinical trial beginning. This is different from any other kind of clinical trial. As with other kinds of drugs, The FDA regulates the quality and safety of gene therapy products and supervises how these products are used clinically. Therapeutic alteration of the human genome falls under the same regulatory requirements as any other medical treatment. Research involving human subjects, such as clinical trials, must be reviewed and approved by the FDA and an institutional review board. History: 1970s and earlier. In 1972, Friedman and Roblin authored a paper in Science titled Gene Therapy for Human Genetic Disease. Rogers, 1970, was cited for proposing that exogenous good DNA be used to replace the defective DNA in those who suffer from genetic defects. 1980s. In 1984, a retrovirus vector system was designed that could efficiently insert foreign genes into mammalian chromosomes. 1990s. The first approved gene therapy clinical research in the U.S. took place on 14 September 1990 at the National Institutes of Health (NIH) under the direction of William French Anderson. 4-year-old Ashanti De Silva received treatment for a genetic defect that left her with adenosine deaminase deficiency (ADA-SID), a severe immune system deficiency. The defective gene of the patient's blood cells was replaced by the functional variant. Ashanti's immune system was partially restored by the therapy. Production of the missing enzyme was temporarily stimulated, but the new cells with functional genes were not generated. She led a normal life only with the regular injections performed every 2 months. The effects were successful, but temporary. Cancer gene therapy was introduced in 1992/93 Trojan et al., 1993. The treatment of glioblastoma multiforme, the malignant brain tumor, whose outcome is always fatal, was done using a vector-expressing antisense IGF RNA clinical trial approved by NIH Protocol No. 160224 November 1993 and by the FDA in 1994. This therapy also represents the beginning of cancer immunogene therapy, a treatment which proves to be effective due to the anti-tumor mechanism of IGF-I antisense, which is related to strong immune and apoptotic phenomena. In 1992, Claudio Bordignon, working at the Vitas Salute San Rafael University, performed the first gene therapy procedure using hematopoietic stem cells as vectors to deliver genes intended to correct hereditary diseases. In 2002, this work led to the publication of the first successful gene therapy treatment for ADA-SID. The success of a multi-center trial for treating children with SID severe combined immune deficiency or bubble boy disease from 2000 and 2002 was questioned when 2 of the 10 children treated at the trial's Paris center developed a leukemia-like condition. Clinical trials were halted temporarily in 2002, but resumed after regulatory review of the protocol in the US, the United Kingdom, France, Italy, and Germany. In 1993, Andrew Gobia was born with SID following prenatal genetic screening. Blood was removed from his mother's placenta and umbilical cord immediately after birth to acquire stem cells. The allele that codes for adenosine deaminase ADA was obtained and inserted into a retrovirus. Retroviruses and stem cells were mixed, after which the viruses inserted the gene into the stem cell chromosomes. Stem cells containing the working ADA gene were injected into Andrew's blood. Injections of the ADA enzyme were also given weekly. For 4 years, T cells, white blood cells, produced by stem cells, made ADA enzymes using the ADA gene. After 4 years, more treatment was needed. Jess Gelsinger's death in 1999 impeded gene therapy research in the U.S. As a result, the FDA suspended several clinical trials pending the reevaluation of ethical and procedural practices. 2000s. The modified cancer gene therapy strategy of antisense IGF RNA NIH and at 1602 using antisense/triple helix anti-IGF-I approach was registered in 2002 by Wiley Gene Therapy Clinical Trial and at 635 and 636. The approach has shown promising results in the treatment of 6 different malignant tumors: glioblastoma, cancers of liver, colon, prostate, uterus, and ovary. Collaborative NATO Science Program on Gene Therapy USA, France, Poland, and at LST 980,517 conducted by J. Trojan et al. This anti-gene antisense/triple helix therapy has proven to be efficient due to the mechanism stopping simultaneously IGF-I expression on translation and transcription levels, strengthening anti-tumor immune and apoptotic phenomena. 2002. Sickle cell disease can be treated in mice. The mice, which have essentially the same defect that causes human cases, used a viral vector to induce production of fetal hemoglobin HbF, which normally ceases to be produced shortly after birth. In humans, the use of hydroxyurea to stimulate the production of HbF temporarily alleviates sickle cell symptoms. The researchers demonstrated this treatment to be a more permanent means to increase therapeutic HbF production. A new gene therapy approach repaired errors in messenger RNA derived from defective genes. This technique has the potential to treat thalassemia, cystic fibrosis, and some cancers. Researchers created liposomes 25 nanometers across that can carry therapeutic DNA through pores in the nuclear membrane. 2003. In 2003, a research team inserted genes into the brain for the first time. They used liposomes coated in a polymer called polyethylene glycol, which, unlike viral vectors, are small enough to cross the blood-brain barrier. Short pieces of double-stranded RNA, short interfering RNA or cRNA, are used by cells to degrade RNA of a particular sequence. If a cRNA is designed to match the RNA copied from a faulty gene, then the abnormal protein product of that gene will not be produced. Gendecine is a cancer gene therapy that delivers the tumor suppressor gene 53 using an engineered adenovirus. In 2003, it was approved in China for the treatment of head and neck squamous cell carcinoma. 2006. In March, researchers announced the successful use of gene therapy to treat 2 adult patients for X-linked chronic granulomatous disease, a disease which affects myeloid cells and damages the immune system. The study is the first to show that gene therapy can treat the myeloid system. In May, a team reported a way to prevent the immune system from rejecting a newly delivered gene. Similar to organ transplantation, gene therapy has been plagued by this problem. The immune system normally recognizes the new gene as foreign and rejects the cells carrying it. The research utilized a newly uncovered network of genes regulated by molecules known as microRNA. This natural function selectively obscured their therapeutic gene in immune system cells and protected it from discovery. Mice infected with the gene containing an immune cell microRNA target sequence did not reject the gene. In August, scientists successfully treated metastatic melanoma in 2 patients using killer T cells genetically retargeted to attack the cancer cells. In November, researchers reported on the use of VRX-496, a gene-based immunotherapy for the treatment of HIV that uses a lentiviral vector to deliver an antisense gene against the HIV envelope. In a phase I clinical trial, 5 subjects with chronic HIV infection who had failed to respond to at least 2 antiretroviral regimens were treated. A single intravenous infusion of autologous CD4 T cells genetically modified with VRX-496 was well tolerated. All patients had stable or decreased viral load. 4 of the 5 patients had stable or increased CD4 T cell counts. All 5 patients had stable or increased immune response to HIV antigens and other pathogens. This was the first evaluation of a lentiviral vector administered in a U.S. human clinical trial. 2007. In May, researchers announced the first gene therapy trial for inherited retinal disease. The first operation was carried out on a 23-year-old British male, Robert Johnson, in early 2007. 2008. Leber's congenital amaurosis is an inherited blinding disease caused by mutations in the RPE65 gene. The results of a small clinical trial in children were published in April. Delivery of recombinant adeno-associated virus AAV carrying RPE65 yielded positive results. In May, two more groups reported positive results in independent clinical trials using gene therapy to treat the condition. In all three clinical trials, patients recovered functional vision without apparent side effects. 2009. In September, researchers were able to give trichromatic vision to squirrel monkeys. In November 2009, researchers halted a fatal genetic disorder called adrenoleukodystrophy in two children using a lentivirus vector to deliver a functioning version of ABC-DUN, the gene that is mutated in the disorder. 2010. An April paper reported that gene therapy addressed achromatopsia color blindness in dogs by targeting cone photoreceptors. Cone function and day vision were restored for at least 33 months in two young specimens. The therapy was less efficient for older dogs. In September, it was announced that an 18-year-old male patient in France with beta thalassemia major had been successfully treated. Beta thalassemia major is an inherited blood disease in which beta hemoglobin is missing and patients are dependent on regular lifelong blood transfusions. The technique used a lentiviral vector to transduce the human B globin gene into purified blood and marrow cells obtained from the patient in June 2007. The patient's hemoglobin levels were stable at 9 to 10 g/dl. About a third of the hemoglobin contained the form introduced by the viral vector and blood transfusions were not needed. Further clinical trials were planned. Bone marrow transplants are the only cure for thalassemia, but 75% of patients do not find a matching donor. Cancer immunogene therapy using modified antigen, antisense/triple helix approach was introduced in South America in 2010/11 in La Sabana University, Bogota Ethical Committee 14, December 2010, No P-4-10. Considering the ethical aspect of gene diagnostic and gene therapy targeting IGF-I, the IGF-I-expressing tumors, i.e. lung and epidermis cancers, were treated. Trojan et al., 2016. 2011. In 2007 and 2008, a man, Timothy Ray Brown, was cured of HIV by repeated hematopoietic stem cell transplantation. See also allogeneic stem cell transplantation, allogeneic bone marrow transplantation, allotransplantation with double delta-32 mutation which disables the CCR5 receptor. This cure was accepted by the medical community in 2011. It required complete ablation of existing bone marrow, which is very debilitating. In August, 2 of 3 subjects of a pilot study were confirmed to have been cured from chronic lymphocytic leukemia (CLL). The therapy used genetically modified T cells to attack cells that express the CD19 protein to fight the disease. In 2013, the researchers announced that 26 of 59 patients had achieved complete remission, and the original patient had remained tumor-free. Human HGF plasmid DNA therapy of cardiomyocytes is being examined as a potential treatment for coronary artery disease as well as treatment for the damage that occurs. In 2011, Neovasculgen was registered in Russia as the first-in-class gene therapy drug for treatment of peripheral artery disease, including critical limb ischemia. It delivers the gene encoding for VEGF. Neovasculgen is a plasmid encoding the CMV promoter and the 165 amino acid form of VEGF. 2012, 2013, 2014, 2015, researchers successfully treated a boy with epidermolysis bullosa using skin grafts grown from his own skin cells, genetically altered to repair the mutation that caused his disease. 2016, In April, the Committee for Medicinal Products for Human Use of the European Medicines Agency endorsed a gene therapy treatment called Strimvelis, and the European Commission approved it in June. This treats children born with adenosine deaminase deficiency and who have no functioning immune system. This was the second gene therapy treatment to be approved in Europe. In October, Chinese scientists reported they had started a trial to genetically modify T cells from 10 adult patients with lung cancer and re-inject the modified T cells back into their bodies to attack the cancer cells. The T cells had the P-D-1 protein, which stops or slows the immune response, removed using CRISPR-Cas9. A 2016 Cochrane systematic review looking at data from 4 trials on topical cystic fibrosis transmembrane conductance regulator CFTR gene therapy does not support its clinical use as a mist in hay. One of the 4 trials did find weak evidence that liposome-based CFTR gene transfer therapy may lead to a small respiratory improvement for people with CF. This weak evidence is not enough to make a clinical recommendation for routine CFTR gene therapy. 2017. In February, Kite Pharma announced results from a clinical trial of CRT cells in around 100 people with advanced non-Hodgkin lymphoma. In March, French scientists reported on clinical research of gene therapy to treat sickle cell disease. In August, the FDA approved tisagenlecleucel for acute lymphoblastic leukemia. Tisagenlecleucel is an adoptive cell transfer therapy for B-cell acute lymphoblastic leukemia. T cells from a person with cancer are removed, genetically engineered. The T cells are engineered to target a protein called CD19 that is common on B cells. This is the first form of gene therapy to be approved in the United States. In October, a similar therapy called axicabtagene ciloleucel was approved for non-Hodgkin lymphoma. In October, biophysicist and biohacker Josiah Zayner claimed to have performed the very first in vivo human genome editing in the form of a self-administered therapy. On 13 November, medical scientists working with Sangamo Therapeutics, headquartered in Richmond, California, announced the first ever in-body human gene editing therapy. The treatment, designed to permanently insert a healthy version of the flawed gene that causes Hunter syndrome, was given to 44-year-old Brian Maddox and is part of the world's first study to permanently edit DNA inside the human body. The success of the gene insertion was later confirmed. Clinical trials by Sangamo involving gene editing using zinc finger nuclease ZFN are ongoing. In December, the results of using an adeno-associated virus with blood clotting factor Roman 8 to treat non-hemophilia patients were published. 6 of the 7 patients on the high-dose regime increased the level of the blood clotting Roman 8 to normal levels. The low and medium dose regimes had no effect on the patient's blood clotting levels. In December, the FDA approved Luxturna, the first in vivo gene therapy, for the treatment of blindness due to Leber's congenital amaurosis. The price of this treatment is $850,000 for both eyes. 2019. In May, the FDA approved onasemogene abeparvovic Zolgensma for treating spinal muscular atrophy in children under 2 years of age. The list price of Zolgensma was set at $125 million per dose, making it the most expensive drug ever. In May, the EMA approved betiblogene autotemsyl Zynteglo for treating beta thalassemia for people 12 years of age and older. In July, Allergan and Editas Medicine announced phase I/Roman 2 clinical trial of AGN-151,587 for the treatment of Leber congenital amaurosis 10. This is the first study of a CRISPR-based in vivo human gene editing therapy, where the editing takes place inside the human body. The first injection of the CRISPR-Cas system was confirmed in March 2020. 2020s. 2020. 2021. In May, a new method using an altered version of the HIV virus as a lentivirus vector was reported in the treatment of 50 children with ADA-SID, obtaining positive results in 48 of them. This method is expected to be safer than retroviruses, vectors commonly used in previous studies of SID where the development of leukemia was usually observed and had already been used in. This was done in vivo without taking cells out of the patient to edit them and reinfuse them later. In July, results of a small gene therapy phase I study was published reporting observation of dopamine restoration on 7 patients between 4 and 9 years old affected by aromatic L-amino acid decarboxylase. 2022. In February, the first ever gene therapy for Tay Sachs disease was announced. It uses an adeno-associated virus to deliver the correct instruction for the hexad gene on brain cells, which causes the disease. Only 2 children were part of a compassionate trial, presenting improvements over the natural course of the disease and no vector-related adverse events.